It is in continuation of my uh, previous video which I have detailed about the process. Frederick uh, Frederick Sanger, who have devised this DNA sequencing technique, and this technique was called. Presently, it is not very useful because method, chain termination method by dideoxynucleotides by dideoxynucleotides is replaced by the method used by Maxam and Gilbert, Maxam and Gilbert, who have used this as a method of DNA sequencing. So Alan Maxworth and Gilbert, they have made this technique in 1976 to 77, and this method is bit chemical method for DNA sequencing, chemical method for DNA sequencing. So it is very interesting because this same thing, the previous the DNA sequencing has many implications. DNA sequencing is very useful because we have a targeted genome sequencing we want some every gene in a every gene and the nucleotide order to be in a determined form, so that we can combat, or we can we can find out several medicine we can find out several medicine, and we can find out several we can correct several genetic diseases also, genetic diseases. Then you can use it in the mutation, determination of mutation, how which part of the gene, which gene is mutated, it can be sequenced. Then you can use it for evolutionary evolutionary uh, relation, find out the evolutionary relationship among the various form of various organism group or phylum. It is also useful. So, and you can use it in the DNA fingerprinting also, fingerprinting. And nowadays it is used in criminal, that means detection. So these are the several things can be done by DNA sequencing, or it is based on, that means like it is very short, that is not so time consuming, and it is very much that means perfect method, and it is done by Maxam and Gilbert. So this chemical modification is, once again, it is 5 prime to 3 prime, and here is DNA 3 prime to 5 prime. This is called your DNA, and this DNA molecule is two double stranded, and they are complementary to each other. But first of all, when we do this thing, we follow few steps. What are these steps? We can denature the same process, the DNA into single stranded form, and this single stranded DNA, and now you can have several pieces of single stranded DNA, and then it is fragmented. Fragmentation can be done by using restriction endonucleases. So restriction endonuclease, it is site specific, and it cut out the DNA molecule into various pieces. Sequencing these genes, or you how will you find out the proper order? See, this is you, all are genes. Suppose this gene has this sequence in order, nucleotides order, or suppose this has this order. So it is maybe several type. So you have to find out this actually how the order is arranged. I was this for this sample DNA I am taking one DNA. So after this step, this two step, suppose I have one DNA strand after restriction endonuclease digestion, single strand I have an sequence a, suppose G T T, uh, then C T T A C G. This is the sequence. Now here you see I have given on. So it is chemical basis. So you have certain chemical molecules which will help you. So A plus G, then you can C plus T, C plus T, or C. These are the four. So now these are there are certain chemicals like the dimethyl sulfonate or dimethyl sulfate, piperidine, dimethyl methyl sulfate plus piperidine. These are chemicals. If you use this thing, whenever there is G, it will eliminate this out, or it will cleavage this G. It is removal of G, removal of G from this sequence. So whenever there is G, that will cut down. So you will see if I put some G's in between. So G. So now you see what how you will find some, because one is forming, say up to this. So A G T T G C T T G T A C. So this much we will get. So one big strand. Say again, this is your first cut. So again another, when again you will put, so this time this up to this. So suppose 1 2 3 4 5 6 7 8 9 10 11 12 13. So you will get 1 2 3 4 5 6 7 8 9 50 10 11 12 13. So another cut will be done between 7 and 7. So up to 7 you will get another strand. Here you have got up to 12, 7. Again another thing is up to 4. So here you will get up to 4. So it is very scientific. You can get this up to 4. So 4 nucleotide number. Please, if you don't follow this one, you will never understand how this chemical digestion help to determine the sequence. And now suppose you have changed the sample with chemical. You have changed the sample with other, that means dimethyl sulfate DMS plus piperidine with formic acid. If you are in a big lab, everything is available, your guide will provide everything. So here, so this, it can remove either G or A. This is the trick here, because this is the thing. It can remove either A or G, any one of them from this. So you will see now you A and G. So if it remove AG in the sequence, suppose CG, so here you will get another 12. G is removed, so you will get 12, 12, 13. So you will get 12 up to 12. The total nucleotide number is 12. 12 numbers nucleotide. Then you suppose G and A and G, suppose here you can get, here you can get, you will get suppose 9 nucleotide band. You will get uh, 9 nucleotide band. You can get yes, 9 nucleotide band. You can get 10 nucleotide band. So these bands will come again. You can get, so yes, 2 band will be here. And if you get C and T here, you will get C, yes, you will get a band of 11, a band of 11. Again C and T, find out here, band of 6 you will get. You can get band of 6. You can get band of 8. Okay, so if the cut is here, so 8. So in this way you will get for C also, if you find out the band. So now every band will there, and now you prepare wells. Now you prepare a band of, if you prepare a gel, same gel, and you do not need any primer. What I have discussed in previous, you do not need any primer. You do not need primer, DNA polymerase, nothing is required in this case. So nothing is required. So now what is very important, you need the separate gel electrophoresis is required. Gel electrophoresis, DNA gel electrophoresis, DNA gel electrophoresis. So now you will run G here, then A plus G vowel here, then you run C plus T here, and lastly you set the last one, it is your C. It is your C. Here you see you have 4 things. It is your C. So if you put the C1 here, so now you see gel, and now you have from any one of this, you can understand. If you think J, so it is, this depends on size, total kilodaltons they have, so it will move in separal distance, and it is radio level. So the sequence short, that means here I have shown you 13 nucleotides, so you will get many bands. It may be more than more band, that means, so it is the final sample solution will show these bands common, and then you will get several band. Again you remember, sometimes what happen, you will find same bands, sometimes you will get same bands. So if you get same rank, that means if you think this row or column, you are getting these two bands. So now you have to give value when this bending is done. You will get G for these two, A plus G. You give only G, you take, and then you can do another thing. If you will find several one bands and two bands, so ultimately you will get 13 ranks. Ultimately you will get 13 rank. Please be sure that your bands are 13. So these 13 bands, from 13 bands you will do the calculation, and you will get, you just progress this way. That means from lower to higher. So if you follow this lower to higher, that means numbering, that means A, T, G, C, whatever is given, you can easily you take the smaller bands from lower to upper. You take smaller bands in the 5 prime direction. So it is 5 prime and you are progressing up. So larger width, because this is the 3 prime end with the large. So because you see you are cutting this way, so this is 3 prime. So you consider each of these nucleotides from these horizontal bands. You will find a single one. Let me show you one. Suppose you are getting in this particular row, in this rank, you will not find anything. Or if you get these two, I have told you the trick. You take G and A plus G. So you will take G, and same thing. So hope this chain termination method what I have shown you will help you to find out several problems in CSR and all. So sequencing by Maxam and Gilbert, the nucleotides in my that means sample I have shown here. Accordingly, in my example I have did. So the question what we get in the CSIR and all, so you will get this, let me tell you the question that is given during the exam. They asked, they provide this chart, and what you have need to study very carefully what actually they are telling. The question you go through repeatedly, because it is one of the very interesting part, and student can avail answering from this part. So please read out this thing very carefully, and definitely whatever I have given you as a material, it will help you definitely. Whatever the question, sometimes it is linked with evolution and all. In that case you need a integrated knowledge of the thing. So thank you, have a nice day. Hope for your success. So it is, the video is specifically made for CSR net, CSR net in life science, whatever your scheme may be, or it can be used for ICR net in agricultural biotechnology. Thank you, have a nice day.